Everywhere we look in nature, we find enormous diversity. You need only look at the difference between Adrian and I to see proof of this. How you doing? But why should this be? Why are we not just cell-for-cell, uniform copies of one another? Well, it has to do with disease.
If you ask a plant biologist, they will tell you about something called the gene-for-gene model. This model predicts that recognition leads to resistance. This is to say that for a specific pathogen genetic type, hosts that have matching types in their genome will be able to mount a defense and stop the spread of infection. Gene for gene, GFG. Each host parasite genetic pair is an opportunity for resistance. The parasite gene can be either virulent or avirulent. The virulent gene is universally effective. The host gene can be of two types also, resistant or susceptible. The resistant gene can mount a response when presented with its avirulent pair. When a GFG pair between a resistant host gene and an avirulent parasite gene occurs, the infection is met with resistance.
If you were to ask an invertebrate biologist, however, they would probably tell you about the matching allele model. An allele is an alternate version of a gene. Not necessarily better or worse, just different. Take hair color. I have the allele for brown hair, but you may have the allele for blonde or even ginger hair. The rule for this model is simpler than GFG. A pathogen can only infect a host if their alleles perfectly complement each other. Think of it like a lock and key mechanism, where only one particular key will open one particular door. It is now generally believed that gene for gene and matching allele models are not opposite sides of a coin, but at opposite ends of a spectrum, where it's possible for all manner of subtle host and parasite interactions to exist.
As discussed, GFG is used by plant biologists, frequently to model agricultural interventions. The classic example of GFG interaction, and the one we saw in lecture, is by the original GFG proponent, Harold Henry Flor. He identified one pair of matching resistance and avirulence genes in rust of flax infections. We'll present a more modern case study that has yet to be covered in class. Our GFG case study takes us on a journey across the world, where we settle on a generic rice paddy in rural Asia. We are here to examine the relationship between Oryza sativa, that's to say common Asian rice, and the fungal pathogen Magnaporthy griseae, based on research by Gia and colleagues in the year 2000. M. griseae is a widely studied, highly virulent fungal pathogen present everywhere rice is grown. The amount of rice it destroys each year could feed 60 million people. It is a well-documented gene-for-gene interaction with O. sativa. Rice gene PETA confers resistance to rice blast fungus. It encodes for a cytoplasmic receptor. Fungus gene AVR PETA encodes for a ligand that directly binds to the PETA receptor. This is the receptor-ligand model of GFG interactions, where the R and AVR products directly interact with one another to enable resistance.
Matching allele models, or MAMs, fundamentally differ from GFG models. Quite simply, universal virulence is just not possible in an MAM model, and polymorphism can be easily maintained. But what about the proof for these MAMs, which constitute a central tenet for the Red Queen hypothesis? A great deal of empirical evidence for matching allele models have been generated by studying Daphnia magnia populations and its parasite, Pasteuria ramosa. Much of this research was conducted by a group headed by a Dutchman named Luix, who published a series of related studies between 2010 and 2016. They tested Daphnia genotypes for resistance to Pasteuria ramosa, and they observed that different alleles at the same locus can select for specific genotypes of a pathogen. In all of this, they did not find any host clone that was resistant to all parasites, or any parasite genotype that was infective in all host clones. They found only that the host clones of Daphnia were either completely susceptible or completely resistant to the parasite. Fortunately, someone other than Luix's group had something to say about matching allele systems. A study by Khalifa and colleagues was investigating potato virus Y pathogenicity against peppers, discovering that infection depended on the direct binding between specific proteins between the virus and the pepper, the disruption of which prevented the infection from taking hold. What is this, if not evidence for the matching allele models?
In 2002, Agrawal and Lively proposed a new model that suggested that gene for gene and matching allele hypotheses were not separate models, but rather two ends of the same continuum. By assuming that there was a cost for parasite resistance with the host and cost for virulence in the parasite, they were able to make gene for gene models with matching allele character, because it would prevent any single host from being the best against all parasite genotypes. This has been a BioU 8 ID report. We've been Angus and Adrian. Thank you for watching, and good night.